Hi, let's review together what we learned at gastrointestinal motility. If we think why our intestine moves, you need to remember that the digestion starts at the mouth, and the breaking of the food starts at the mouth, but it's really completed then later on in the GI tract, in the gastrointestinal tract. Now the intestine, the stomach, are able to digest the food first by the enzymes, but without the movement, without the ability to run the food back and forth, then there will be no digestion whatsoever. The other advantage of the movement, other than moving the bowel from one end to another, is allowing the food to mix with the juices, to mix with the enzymes, and also allow the food better contact with the GI wall for the absorption, for the maximum absorption. So you want things to be constantly in moving, and as you can see here in the slide, here are the functions or the benefits of having motility or muscular contraction in the GI tract. You will realize that it moves the ingested matter through, so it moves from one tube, from one end to another end. That's understandable, but important also, it mixes the ingesta with the digestive juice. It also helps in the breaking of the food by running the food back and forth. It maximizes the interaction between the ingesta and the GI tract. It causes mechanical reduction. It reduces the food into a manageable size for your GI tract. Okay, so these are the function and why do we have motility. And I assume if you get a question, it will be an exception question. So we'll give you which of those is not attributed to the mobility, and we'll give you 3 or 4 correct answers from this slide, and we'll give you one wrong answer that the motility has nothing to do with it. Okay, so moving along, if we look at the GI histology, we will realize that it's really common in all the layers, in all the levels, starting from the esophagus all the way to the rectum, with some exception. For example, like we talked earlier, the esophagus has skeletal muscles. Another exception would be the stomach has an additional oblique layer. A third exception would be the GI tract at the very end in the large intestine. The longitudinal fibers are not continuous layer. It broke into straps, and we call those 3 straps tinea coli, if you remember. So we're going to review that again, but let's look at the classical or the generic structure of the GI tract. We understand that we will have mucosa, submucosa, muscularis, and then adventitia or serosa, based on whether or not there is a peritoneum. So if we think of the mucosa, it's going to vary. So the epithelium can be stratified squamous, it can be simple columnar, it can be columnar epithelium with villi. So it varies a little bit depending on or based upon which level are we talking about. That's the epithelium covering. Immediately underneath the epithelium is a very thin layer of connective tissue, areolar connective tissue, and we call that lamina propria. Lamina propria. And underneath that, we're going to have a very thin muscle layer, and we can't even see it. It probably is this one here. And that will be the muscularis mucosa. Muscularis mucosa. Sounds good? So all of that will be consisting of the mucosa. What you see here is the submucosa, and in the submucosa you will have blood vessels, you will have lymphatics, and nerve fibers. All of that you will see here in the submucosa, although there are also some nerve fiber plexus we will see here later on at the muscularis layer. Getting to the muscularis, we will have inner circular, outer longitudinal. Generally it is this way, again with the exception of the esophagus, the stomach, and the large intestine, it's broken into 3 straps. So the muscularis externa, or the muscularis layer, it will have smooth muscle fibers. These are autonomic fibers. You don't have control over it, but we will see later on that there is myenteric plexus, which really controls the movement of your smooth muscle fibers in the intestine, and we'll talk about that in the next 2 slides. But let's just appreciate for now the layers. Let's go over it again. We have the mucosa, and there will be epithelium, and we have lamina propria. You can hardly see it. It's just little connective tissue immediately underneath the epithelium. And then immediately following that will be a very thin layer of smooth muscle fibers, and we call that muscularis, and because it belongs to the mucosa, so we call it muscularis mucosa. And then under that we're going to see the mucosa layer, and we agreed that we will have the lymphatics, the blood vessels, and nerve plexus will be around here in the mucosa. Under that, or outside that rather, we will have the muscularis externa, and generally we have 2 layers, inner circular, outer longitudinal, and then finally we will have either the serosa or the adventitia if the tract is covered with peritoneum, then we will call it serosa. If it's not covered with peritoneum, then we will call it adventitia. Sounds good? So we move on to the innervation of the GI tract. I want you to realize that the smooth muscle fibers, also the glands, all the secretory cells in the GI tract will be innervated by the following: autonomic nervous system, as we land in the nervous system from the sympathetic and the parasympathetic fibers. Endocrine system will have a lot of hormones that directly impact your GI motility. And paracrine agents, one cell is talking to the other, and we will see that, for example, with the gastrin secretion from your stomach, but it's also affecting the stomach. And the central nervous system, we know if we are nervous or if we're angry, then the movement is more. We know also that angry people get more ulcers because of the stress level and the hypersecretion of acids and so forth. So these are the things that control our smooth muscle fibers and the gland secretion: autonomic nervous system, endocrine system, paracrine agents. Paracrine are cell talking to the neighboring cell, and central nervous system. Now, the enteric nervous system is very, very, very important, and we have 2 plexus. One of them is between the 2 layers of the smooth muscle fibers. We call that myenteric plexus of Auerbach. So how do I remember it? First of all, it's myenteric, so it has an M. It controls the motility, so it also has an M, and it's between the 2 layers of the muscle fibers. Okay, so the myenteric plexus controls the motility of what? Of the muscularis. Where is it at? Between the 2 layers of the muscularis externa. It regulates the motility, and by regulating the motility you will be able to change the rate, intensity, and the velocity of the contraction. Just remember, the myenteric deals with muscles. Very different than the Meissner. The Meissner, the submucosal plexus of Meissner, these guys deal only with the secretion. Deal with the secretion and the absorption, in and out. And where are they at? They will be in the submucosa. Sounds good? So the submucosal plexus of Meissner will control the absorption and the secretion, and the myenteric plexus of Auerbach control the motility, and it is between the 2 layers of the muscularis externa. We need to remember that. My guess is there will be questions regarding this particular point. Okay, so here we have the outer longitudinal fibers and the inner circular, and in between the 2 of them you will see the Auerbach or the myenteric plexus. Over here we will see some plexus, and these are collection of nerve fibers, and these will be in the submucosa. If we go back to the anatomy of the intestine, we will see here the muscularis mucosa layer, and here we have the lamina propria, and here we have the mucosa itself, which is in this case is columnar epithelium. Sounds good? So moving along, what kind of motility do we have? Well, we have mixing the food together, we have segmentation, and in segmentation you break up the contents onto sort of like you have a tube balloon and you make segments in it, and that allows for the breaking of the food, mixing, digestion, absorption. Peristalsis movement, this is how a snake swallows a rat. It's wave-like contractions that propel the chyme or chyme from one end to another, and it pushes the food into one direction, and that's what we call peristalsis. The opposite of it, and you will see that, for example, in the colon, when we get to it, we have anti-peristalsis, so the peristalsis going in the opposite direction. Clearing, that's for emptying your GI tract, emptying the stomach, emptying the colon, even emptying the small intestine, which empties into the large intestine at the ileocecal valve. Retrograde motility, when the movement goes backwards, and that again rocks the food back and forth to allow better digestion, and better absorption, so you don't miss a chance. If the food is moving too fast, and that happens when we have diarrhea, the problem is we have inflammation and the food keeps moving fast, and if it moves fast, then there is no chance for adequate absorption. Sounds good? So we go to the smooth muscle fibers. All over the GI tract you will have the contraction. We agreed that we have sympathetic innervation. The sympathetic innervation tells your intestine it's not really the time now to spend the energy into absorption and digestion, so it slows down the movement. The sympathetic slows down the movement of the GI tract. The parasympathetic is the wine and dine system, where you will increase the secretion, increase the sympathetic, increase the peristalsis, and increase the digestion, increase the absorption. That will be with the parasympathetic innervation. Sounds good? So if we talk a little bit about the esophageal muscularis externa, it's a little different, because at the upper 1/3 you will have a mixture between skeletal muscles and smooth muscle fibers that allows you some degree of voluntary action when you are swallowing. And all the way down near in the lower 1/3, of course, this becomes completely smooth muscle fibers, which is completely autonomic. Now we have two sphincters. One of them is all the way up, and we call that the upper sphincter, and that doesn't allow the air to come in, so you don't end up swallowing too much air and filling the stomach with air. And then there is a lower sphincter in the esophagus, and that protects against the retrograde movement from the stomach back into your esophagus. And failure, of course, to do that can cause reflux, especially the lower we're talking about, which is your cardia opening. Reflux esophagitis can lead to ulceration of the esophagus and the change of the epithelial lining of the esophagus, and all of it because of the lower sphincter is not closing. Now, what about the upper sphincter? If it's not relaxing, well, then you have something in your throat and you're not able to swallow it. It's you're choking because it's the sphincter all the way up in the upper esophagus is not letting go. Okay? So moving along, sometimes, of course, foreign objects can be swallowed into the esophagus, and especially with kids, also with animals. Sometimes they have a toy or a pin that they swallowed, and it's not too difficult to extract it either from the esophagus itself or if it goes down to the stomach. Okay. That leads us to the stomach. What does the stomach do for us? Well, first it temporarily holds the food, starts the digestion, hydrochloric acid, enzymes like pepsin is secreted. It releases the content little by little, because otherwise this huge meal you ate will go straight to the duodenum or to the ileum, and it's going to distend the ileum or the duodenum or the jejunum rather, or the duodenum into a very large size. So it allows you a slow release of the content, and that will be of course by the cardiac sphincter. Because of the acid and because of the proteolytic enzymes that are secreted by the stomach helps you in clearing up infection, destroying bacteria. Most of the bacteria at least will be destroyed at that level. So let's look at the slide here. So the functions of the stomach would be temporary storage, mixes the food with the gastric secretion, and the gastric secretion will allow for both first of all, the gastric secretion will be chemical degradation, but the mixing itself, the shrunning of the food, that's why we have 3 muscle layers there to ensure that we are having a really good digestive movements going around in the stomach. Liquefaction of the content, because if you remember we talked about it, that the stomach secretes about 1.5 to 3 liters of hydrochloric acid, and that makes the content of the stomach, the sandwich we eat, makes it very liquidy. And when it's liquidy, first it's easy to pass through the intestine, but it's also easy to digest and easy to absorb from, rather than having a very thick and non-fluidy content or solid content. So the liquefaction of the content will happen in the stomach, gradual regulated release of the content into the duodenum, and bacterial and viral disinfection. Okay, so we did talk about this. Moving along, here are the anatomy. Here's the anatomy. It's an open stomach here, and we see that we have the esophagus, and the marcation between the stomach and the esophagus has what we call the cardiac notch, which will be here at the area we call the cardia, which is the junction between the esophagus and the stomach. Now, if we make a line there, anything above that line will be the fundus, the rest will be the body. Going all the way down will be the pylorus, and the pyloric sphincter will be regulating, which is an increase of the smooth muscle fibers to form a sphincter that allows for the slow and the gradual release of the food from the stomach to the intestine, and it also prevents the retrograde movement of the duodenal content back into your intestine. Okay, your mucosa is folded, and we call that the gastric folds or the rugae, and you need to remember that word, the gastric rugae. Okay, and yeah. So the gastric motility, there are 2 sphincters. One of them is between the esophagus and the stomach, and we call that your cardiac sphincter or lower gastric sphincter, and the other one between the stomach and the duodenum, and we call that pyloric sphincter. And why do we have this sphincter? Well, again, we need 2 to 3 hours for the enzymes and the acids to mix with the food before it goes forward. Second, I want a slow release. I don't want everything to go quickly. Third, I don't want the retrograde movement from the duodenum back into the intestine. And that, of course, will be the opening will be promoted by the antral mixing, and the stomach will empty approximately 2 to 3 hours, and then it will go to the duodenum, and that will be the subject of the next slide. So the regulation of the gastric emptying. Why do we have regulation? Because we don't want to overwhelm the intestine. Remember, there are many things that didn't get absorbed in the intestine, in the stomach, such as lipid, for example. So we want the pancreatic lipase to gradually see your chyme. You don't want the chyme to come all of a sudden to your duodenum. It will be overwhelmed, and you don't have enough pancreatic juice to deal with it. Also, the bile salts, which will work into emulsifying your fat, you don't want to overwhelm them. So you will have little by little release from your pyloric sphincter. The intestinal acid is gradually neutralized, and we're going to see that by the bicarbonate. Remember, the problem here is we have hydrogen ions. Once we get into your small intestine, we will have plenty of bicarbonate secretion, and that will neutralize our gastric acidity. So we go into the intestine now. We passed the stomach, we passed the pyloric sphincter, and we go into the intestine. The intestinal structure, again, the generic structure of the intestine, will have the same generic structure we talked about before: mucosa, submucosa, muscularis, and serosa or adventitia. A very small part of the small intestine will be the duodenum, that's about 5%, followed by the jejunum, that's about 40%, and then finally the ileum. The rest of the GI tract, 25% of the GI tract, is the large the large intestine, the esophagus, and the stomach. Okay. Most of the digestion and absorption happens at the duodenum and the jejunum. The jejunum is the place where it has lots of villi, has lots of folding and microvilli, and that will allow more surface interaction with your content. When you go down to the ileum and the rectum, what's more important at that point is the secretion. So you will have more mucus secretion in the distal portion of your intestine. Okay, the jejunum and the ileum, they freely move because they are surrounded by what we call mesentery proper, and the mesentery proper doesn't tie the intestine, the small intestine, to the wall, to the posterior abdominal wall, but rather it will make a fold like I illustrated to you, and that fold will allow sort of free movement of the intestine, of the small intestine, and that's very good when it comes to the motility. All right. If we look at the structure from the inside, we will see that the inside, the first thing that we see are these circular folds, and we call these guys plica circularis. Inside the plica circularis, you will see these villi, these finger-like projections that you can see, and each of those villi will be covered with epithelium. That's columnar epithelium that has also microvilli. So we have plica circularis that slows down the movement, and then we have the villi, and you can see the villi over here, and then we have the microvilli that are brush-like formation on the columnar epithelium itself, and all of that is intended to increase the absorption and increase the interaction between your intestinal wall and your chyme. And proceeding distally, when you go from the jejunum to, for example, the ileum, you will realize that the villi will decrease. We don't need it much over there, but at the same time, because the absorption, most of the absorption already took place in the jejunum, so the content become thicker content, and so we need to lubricate it. So distally we will have more mucus secretion. Proximally we have more villi, which enhances the absorption. Sounds good? We need to remember those facts about the plica circularis, about the villi, about the microvilli. We also need to understand that the more distal you go down, then the less villi and more mucus secretion. That's why we have the goblet cells, which are our mucus secreting cells. Okay, The regulation of the contraction, it is regulated by the enteric nerve plexus, and the amplitude can be affected by both the enteric nerve plexus and the autonomic nervous system. Also hormones. Parasympathetic activation will increase the waves, increase the size, increase the duration. Remember, the parasympathetic stimulation enhances the GI mobility. The sympathetic innervation, because it deals with what we call the fight or flight. You have to increase other systems, but not the intestinal movement. So the parasympathetic slows down the movement. The parasympathetic enhances the movement of your intestine, and the sympathetic would slow down the movement of the intestine. Okay, the kinds of motility we have: peristalsis. Again, we talked about that before. Peristalsis, remember a snake swallowing a mouse or a rat. It is the movement from one end to another that's induced by enteric nervous system reflex, and when the intestine is distended, then we will see the intestinal reflex in a little bit. Distally, you will have relaxation to allow the movement of that bolus downstream. The peristalsis will be controlled by acetylcholine, nitric oxide, and other factors as well. All of these will allow the peristalsis to happen. Now, the mixing is, you want to mix the content of the intestine with the juice, and also bring the center of the bolus or the chyme into the walls of the intestine to allow the maximum interaction. All of that is important for the proper digestion. So as you can see here, mixing, you will be mixing the chyme with the digestive secretion. Also, mixing will facilitate the interaction with the surface of the villi. In segmentation, when the intestinal content is broken into smaller bubbles, and then those bubbles are broken again, this will allow the digestion. Consider that like additional teeth that will break the food back and forth, and segmenting the food into smaller portions. That also will allow the better interaction with the digestive enzymes and the wall of the intestine as well. Okay? All right, these are reflexes. I'm assuming that there might be a question in these guys regarding the reflexes that we have for the intestinal motility. One is called intestinal reflex, and as I explained it for you before, if you have stretching of a proximal region, then the region immediately distal to it is going to relax. That will ensure that once the contraction occurs with the peristalsis, we already have a relaxation distally, which will allow the food to go from one end to another. So once again here, the intestinal reflex is distension proximally causing relaxation distally. Sounds good? Then we have the gastroileal reflex. If you have elevated activity of the stomach, you will accelerate the passage of the chyme through the ileocecal valve to clear the way for the content now that are filling the stomach to replace that. You want to get everything out quickly because you already know that the stomach is full and it has lots of content and lots of activity. So you clear up the ileum by opening the ileocecal valve, and therefore the small intestine will be ready for the next wave of food coming from the stomach. We call that gastroileal reflex. The third reflex is called migrating myoelectric complex, and or controlled by the migrating myoelectric complex, and these are waves of activity, and that happens during fasting, and approximately every 70 minutes or so you will hear that your stomach is growling and that will be propelling the chyme into the colon, or the chyme into the colon, and that's associated with what we call borborygmus, and that is the growling of the GI tract. Okay, so these are the reflexes that we have and the centers that control it. So we have the intestinal reflex, gastroileal reflexes, and the migrating myoelectric complex, which gives you the growling noises when one is hungry. We move from there into the large intestine, but before we get into the large intestine, we will talk a little bit about the ileocecal valve, because it's the junction between the small intestine and the large intestine. The cecum of the colon is separated from the ileum by the ileocecal sphincter, and we agreed that we have many reflexes here, like a gastroileal reflex. When the stomach is full, then this will open up to allow the emptying of the chyme. But you can see here is the ileum over here, and here is the cecum, and here is the ileocecal valve. Okay, so what are the functions of the colon? You will be absorbing water, some salt. It will provide lubrication. We'll have lots of mucus, because now the content of the large intestine is getting thicker, because most of the absorption already happened in the jejunum and the ileum. It will allow for the segmentation of the chyme, and we call that haustration. We will see a picture of the haustra, but you can see here also the haustration. See how it's segmented? It's not a continuous loop. By the way, this is the transverse colon, what you see here, and this is the ascending colon, and this guy here is the descending colon. It completes the digestion, because sometimes we have bacteria, probiotics that will give us benefits like vitamin K. We have probiotics that will regulate our intestinal movement. We have probiotics that helps us overall and improves our immune system. So we have lots of probiotics here, and even in animals that feed on grass, they have intestinal bacteria that allows them to break down cellulose, so they can benefit from eating grass and getting the sugar out of the cellulose. Okay, the process after it's done, then you would like to eliminate the content, and that will be the movements, and that will be done by what we call mass movement, and that's something unique only to the colon. Okay, histology of the colon, we will see lots of goblet. You see all these little bubble cells, these are goblet cells. But other than that, we have the typical or the classical structure. We have the mucosa, we have the muscularis mucosa here, right? So we have epithelium, we have lamina propria, we can't really see it very well. But then we have the muscularis mucosa, and then we have the submucosa, and then we will have the muscularis. The thing about the muscularis is that you will have, yes, the two layers, the circular layer and the outer longitudinal, but the outer longitudinal is not continuous. It's broken into three strands, and we call these straps or strands tinea coli. Tinea coli. Okay, so we see the structure here, and we will see the longitudinal is broken into three bands, and again this is called tinea coli. The motility of the colon is regulated by reflexes. One of them is called colonocolonic, sort of like the same intestinal that we had before. In colonocolonic, if you have distension of the proximal, you will have relaxation of the distal. It's exactly the same reflex that we had before, which we called intestinal. Here we call it colonocolonic reflex. In addition to that, we will have gastrocolic, sort of like the gastroiliac reflex we had. If the food gets into the stomach, so that means we have more activity going on in the stomach, there will be a reflex by your large intestine to empty the content, again to clear up the way. Okay? So, and sometimes we notice that after a meal, then sometimes we feel like going to the bathroom, and that's because the large intestine got stimulated by a gastric reflex. Okay. So, the motility of the colon, just like the motility we learned about before from the stomach and from the small intestine, we're going to have segmentation that's breaking into small bubbles. We have the peristalsis. We also have the antiperistalsis that's going backward, and that slows down the emptying, and that allows more time to absorb the water, absorb the electrolyte. But the unique part about the colon here is we have what we call mass movement, because now the chyme is not liquid anymore, it's semi-solid, so the movement of it, it's almost like labor, moving a large bulk at the same time, and that is strong and prolonged peristalsis. It's not just your regular peristalsis, it's long and very strong peristalsis to allow the movement to kick out the content of the colon as a mass. Sounds good? And haustration, which is the segmentation, this segmentation of the colon is ensured by the structure of the outer longitudinal fibers, which makes what we called tinea coli, and it breaks it into little bubble-like formation, small segments, and we call each of those segments the haustra. Okay? All right, and I don't think you would be asked about the inflammatory bowel disease, but in some people where after some kind of a trigger, this trigger can be aspirin, ibuprofen, infection, or whatever, they have inflammation, they have acute inflammation. Generally, the normal people will have tolerance to that, and you will have complete healing. And in some cases, especially if you don't have good immunoregulation or failure to repair, or you have some consistent bacteria there causing this, and if you have genetic susceptibility, then you will run into chronic inflammation, and that causes diarrhea and ulceration of the large intestine. I don't believe that this is something that we would be asked about, because this is not a pathophysiology course, of course. Anyhow, finally we get into the rectum. And the rectum, the mass movement will fill the rectum prior to defecation, and then there will be a reflex relaxation of the internal anal sphincter if this is stretched. Now, of course, we inhibit this reflex constantly, and we also, the external sphincter of the anus, these are skeletal muscles that we can control, and that we can control our ability to eliminate the food from our rectum, so it doesn't happen autonomically. Defecation is, as I said, is determined then by both a reflex, that's the stretching, and by also voluntary events. And why is it voluntary? Because we can inhibit the reflex, and we can also induce contraction of these skeletal muscles that you can see here, the external anal sphincters, and that doesn't allow the content of the rectum to get out. Okay, the frequency of the bowel movements during normal healthy people, it varies from 3 times a day into 3 times a week, but generally healthy wise it should be about 1 to 2 times a day. The more fibers, of course, the more spicy food a person would eat would allow the better passage and better regulation of the intestinal movement. Okay?